Swayam Prabha. Digital India. Educated India.
So today we will focus on transcription factor, and then we will move on to modifications to DNA itself and how they influence differential gene expression and therefore development. Okay. So transcription factors usually contain three different domains. This does not mean all of them have all the three all the time. So, but by and large most of them have this. A DNA binding domain where actually this is part of the protein that binds to the DNA directly in amino acids interacting with the DNA double helix, and a transactivating domain where other proteins or factors that bind might modulate the activity of that particular transcription factor, and a third one protein-protein interaction. So, where either some of the transcription factors, for example, the example that we are going to see acts as a dimer. So, the two polypeptide chains interact in that region. Sometimes other proteins interact and influence their activity. Okay. So, the transactivating one is the one that is responsible for actually activating or not activating the RNA pol2 eventually. So, and these are, their effect is really dramatic, and therefore we are going to see a real example where the defect causes a disease, just to keep you motivated to listening to this. An example is MITF. So this transcription factor is expressed in ear, skin and pigment forming cells of the eye, okay, like irises. And if you have mutation, you will have problem hearing and multicolored irises and then white forelock, as you see in this picture. See, the mother has white forelock and her daughter also has, genetically inherited. So very specific problems that you have when you have a specific transcription factor missing. So their activity is very important. And so this gives you, going back to those three domains. So in this protein dimerizes, so the middle portion where it is marked, where you have protein-protein interaction domain. So here that helps them to dimerize, and this long carboxy terminal region is the one that helps in recruiting other proteins, for example a histone deacetylase, etc. And the amino terminal region is where the DNA binding domain is located. So this DNA binding domain, there are different types of DNA binding domains, and based on that the transcription factors are classified into several classes, and within those classes small variations in the sequence might define which promoter they bind, which promoter they do not bind. To give you an idea, we will look at some of them as a table which is there in the book, like homeo domain. So that is a particular DNA binding domain that is conserved and that is present in these proteins listed here. Okay, so we will see them, the Hox protein, in detail several lectures later from now. And then some have this helix loop helix, so HLH, and that is present in these transcription factors, and these are their functions listed here in the rightmost column. And then leucine zipper, they help in binding to, they form like zipper like structure based on the leucine presence. It is usually every 7th amino acid will be a leucine in them. And then you have these zinc finger motifs. These were historically discovered much earlier than the others. So these coordinate as zinc, okay, and that helps in interacting with the DNA, and this is again present in these proteins. Scruple, engrailed, these are all originally discovered in Drosophila, and the names are based on the mutant phenotype, okay, and they are expressed in these tissues. And nuclear hormone receptors, they have zinc finger and they are present in the steroid hormone receptors. And then SRY SOX, that is another domain. So, these are the classes based on variations in the DNA binding domain structure. Okay. So, how do they function, like how do they activate or inactivate or control transcription? Oftentimes it is by one of these two or both. One, they recruit histone modifying enzymes. For example, when a transcription factor binds to a particular sequence, then this transcription factor may recruit histone acetylase, okay, or they might recruit a DNA, an enzyme that removes methyl groups, inhibiting methyl groups from histones. And by doing that, they will displace the nucleosome structure and the DNA gets opened up and more accessible for RNA, Pol2 and other transcription factors. So, essentially by altering the modifications on histones, they open up the chromatin. Okay, so that allows transcription. And the second is they stabilize RNA Pol2. Oftentimes RNA Pol2 bound to the core transcription factors as shown in this cartoon, it is not very stable, but when these transcription factors bound to enhancer, when they interact with all these proteins, they make a much more stable initiation complex. Okay, they stabilize RNA Pol2 on promoter, increasing the probability that that will continue to elongate the initiate the elongation phase. Okay. So, in this structure you see the enhancers can be at a great distance, but through protein-protein interaction the DNA can loop like this. Okay. So this explains if it is present within the coding sequence also, in the introns or whatever, or it can be even the downstream sequence. So, these are the general ways, but there are variations for each transcription factor, but this is generically, if you look at it, these are the major ways by which transcription factors help in controlling the rate of transcription. Alright. So, how powerful are they? So, here you are seeing how the digestive enzyme producing part of pancreas called the exocrine cells, they usually produce the digestive enzymes, proteolytic enzymes and so on, and they do not produce the hormones. What hormones are produced by pancreas? Anyone? Insulin? Glucagon? Anything else? Okay, two are good enough. Okay, insulin and glucagon, right? These cells do not produce. So, here this blue is simply showing the presence of DNA in the nuclei. Okay. Now, you express 3 different transcription factors in this. Okay, PDX1. So, this is expressed in the pancreatic lineage starting from the cells that originally required for the intestinal tube formation. In those cells, if some cells express PDX1, they set the pancreatic lineage. And in that, if you have these 2 transcription factors, they become the endocrine cells of the pancreas. Now, here you have taken exocrine cells. This is in an organism, okay, it is not in the in vitro cell culture. So, this is in the organism where in early on when you express these 3 transcription factors, you have insulin producing cells there. So, the insulin is stained here with the red color, and one of these transcription factors is fused to GFP. So, therefore, you see green, and wherever both are there you get yellow there. In this you can see, here you can see, and here and so on. Okay. So, they are so powerful, they can change the fate of a cell, okay, from exocrine fate to endocrine fate. So, they are very powerful. So, of course, now more dramatic things have been done. Okay. So, people have shown by expressing a few transcription factors, any differentiated cell can be actually converted into undifferentiated pluripotent cells. So, that is even more dramatic than this. So, this leads to a few questions. Okay, how do transcription factors themselves get expressed in a tissue specific manner? Okay. So that is the question. So the answer is quite simple, like the stories that people tell. You know, when I was a kid, I had one person who was several years older to me, like when I was in elementary school, this person was in college. So he talked about some game that he plays, and I asked, who taught you this? He said his, you know, PT master. And I said, who taught him? Then his PT master. So I kept on asking and I never got the relevant answer, because the relevant answer is someone first time discovered it. So similarly, why is this transcription factor expressed in endocrine cells? Because another transcription factor activated it. Why is that active only in pancreatic lineage? It's because another one activated it in the endocrine, the endoderm lineage. So that leads us to what is called transcription factor cascades. Okay, so they work in cascades. Example, MBX activates PAC6, PAC6 activates, we saw already, crystalline, you know, in the lens, and insulin, glucagon, somatostatin, this another pancreatic hormone. Okay, so etc. And similarly, MyoD, this muscle specific really powerful transcription factor, you know, that activates myogenin, which activates other genes involved in skeletal muscle differentiation. So on and so forth, you know, like one after the other. So the main concept here is there is a cascade. Okay, then if you follow up the cascade all the way up to the top, then you have something called pioneer transcription factors. These transcription factors can really open up a highly condensed heterochromatin and initiate transcription. Okay, so it need not be already poised for access to proteins. A good example is this PBX. Okay, so it can go and bind to sequences in a highly condensed repressed chromatin. So that is the definition for pioneer transcription factors. And it probably binds to inhibitors bound to that repressed chromatin, but then once this transcription factor binds, it can recruit, like for example MyoD transcription factor, and it will come with other accessory factors which help in really activating the transcription finally, and like open up the place so that this MEF3, MEF2 etc. can come and bind to their respective enhancers and initiate transcription. So these are the pioneer transcription factors. And on top of it, you have proteins like the Drosophila polycomb group complex protein and trithorax. You need not write down, it is there in the book. So these proteins bind to the histone modifications and maintain a memory of this original activation. Okay, memory of meaning when that particular cell whose fate is specified, when that is going to divide within that individual organism, okay, during the ontogenic stage, all the cells descendants of that particular cell, they will all know that I have to keep this region active, this region suppressed. So those are done by those proteins, the polycomb and trithorax group proteins. So for now you do not need to worry, we will see that concept with the DNA modification also in couple of slides. So this is all about transacting factors controlling transcription. So now there is an opposite phenomena also, enhancers like that. There are other DNA sequences that act as negative enhancers, meaning their sequence prevents the spread of an activation activity. Like for example, if an enhancer activates and if it is going to disassemble the nucleosome, so now is that going to spread along the length of the chromosome and adjacent genes will get activated or what? So you do not want that, right? You want that particular factor to be expressed in that tissue, not all genes. So something has to restrict that, and for that you have DNA sequences to which proteins bind, which insulate or restrict these enhancer activities. Okay, so that is what we are going to see next, and they are often called silencers. Okay, silencers. So silencers are like opposite of the enhancers. So here is one example. So here you have a gene called this, not gene, an element called neural restrictive silencer element. So what it does is it binds proteins that protein is expressed in all tissues except neurons. So, as a result, in all the tissues this sequence will be bound by the protein that binds to this, and there the genes that are under the influence of this particular enhancer will not be expressed. So, therefore, the genes downstream of those promoters will be expressed only in neurons, and as a result this is called neural restrictive silencer. So, here is a reporter where instead of the actual gene you have LAGZ, because you can assay the LAGZ encoded proteins activity. So, when you have this silencer sequence adjacent to it, now you find it is expressed only in the central nervous system here in the embryo, 11.5 day old mouse embryo, and if you do not have that silencer element it is expressed everywhere. Clear? So, these do opposite of enhancers, they restrict the influence. Otherwise what will happen is the enhancer effect will not be very specific and restricted to the genes that need to be activated. It will spread and the control will not be really a tight control. You know, adjacent genes may be partially activated, etc. So, the next we go to modifications that happen to the DNA itself. Okay. So, initially we saw that, do not get confused here. Okay. So, we already saw methylation, etc., and that was at the histone proteins, and that affects the chromatin structure architecture, okay, whether it is tightly coiled with the nucleosomes and histone H1 that is bringing all those nucleosomes together into that solenoid structure, or it is going to be opened up, okay, methylation, deacetylation, all that we saw, and there we saw some methylations in H3 tail can actually be activating, right. So, do not forget that, because oftentimes you may be misled by that. You will just automatically assume methylation means inactivation, acetylation means activation. Acetylation, that is true, that generalization is true, but not for methylation. So, now we are going to look at methylations that happen to DNA. So, I told you to perpetuate an active state or repressed state, we have those trithorax and polycomb proteins that bind to the the modified histones. Like for example, if something is acetylated and you want that to be active, these trithorax proteins bind there and they maintain that state of activated state. But very similarly but more robust is the modifications that happen to the DNA, okay, and that modification happens by methylating the cytosine residues, okay. So, you can have a CH3 added, so therefore, you have the fifth base 5-methyl cytosine, okay. So, this matters a lot in regulation. So, here methylation usually means repressed state, like inactive, that gene is not going to be transcribed, and this can be perpetuated through mitotic cell divisions, okay. We will see that how that happens in couple of slides, we will see that. And second, this can have developmental time factor involved in it, okay, like modification happens at some space and time, not all the time, okay. So, a good example of that is shown by the way hemoglobin genes are expressed, the beta globin, if you take how it is expressed, in that you can see a good example. Initially in the early embryo you have this epsilon version of the globin gene is expressed, its promoter is not methylated, okay. Whereas at that time point you have the gamma globin which is usually expressed in the fetus, that is methylated and it is not expressed, and later as the embryo progresses, then epsilon gets demethylated and that becomes active, this gene gets turned on, okay. It was dormant and now it is turned on, while this epsilon globin gene gets turned off. And when the infant starts to grow, what happens is even this gamma globin gets methylated and inactivated, and beta globin gene gets activated, and that is what is expressed in our body. So, in all of our genome you have the sequence epsilon and gamma sequence, but they are methylated and not expressed. They were expressed sequentially during your embryonic and childhood development. So, now you have only beta globin activation, and these have consequences. Okay, if there is a problem with this, you may have heard this disease thalassemia, that results from failure in the sequential methylation demethylation. So, here what happens is, in these patients you may have problem activating the beta globin. Let us say you have a mutation in beta globin, and now you do not have a functional globin protein produced, although you have perfectly good copies of the gene in the chromosome, but unfortunately they are methylated. So, the methylation program has not been affected, they are active and methylated, but the gene is not expressed although you have wild type copy, and that is how you get this beta thalassemia when it is beta globin gene that is involved. So, this is a very well characterized congenital disease in India, particularly some pockets bordering Andhra and Tamil Nadu, that area, in certain communities of communities where you have marriage among close relatives, like first cousin marriage, or sometimes an uncle marrying a niece, elder sister's daughter, that sister's brother marry. Those are not uncommon. Maybe they are uncommon now, but couple of generations ago they were not uncommon. In those families what happens is, like for example, this given person's sister may be heterozygous, and this guy also may be heterozygous because they come from the same parent and they survived because they are heterozygous. Now there is 1/4 chance their child will be homozygous for the mutant allele. So, that is how you have beta thalassemia running in families, okay, and the underlying cause is this methylation issues. So, now how do you perpetuate this? So, that is where we are getting to. So, usually these methylation block transcription by preventing transcription factors binding to enhancer. Sometimes it is the opposite as well, like some inhibitors will bind to the unmethylated one and they will not bind methylated. Okay. In this particular cartoon what we have is this sequence. Okay. So, you have CG coming together. So, this is often called CPG islands, and its significance will become clear in couple of slides. So, for now do not worry, you just think that this promoter region is usually subject to methylation demethylation. So, when it is not methylated, a transcription factor binds and activates transcription from the downstream promoter. Okay. And if it is methylated, this transcription factor does not bind, as a result the gene is not active. Okay. So, therefore, here this example shows you that DNA methylation blocks transcription factor binding to an enhancer. Okay. And another way by which they function is this methylated cytosine may recruit a protein, like in this case MECP2, which can do two things. One, remove the acetylation mark by recruiting a histone deacetylase, and recruit a histone methyltransferase and mark histones with inhibitory methyl groups. Okay. And due to these two actions, these methylated promoters end up blocking transcription. This sort of methylation based transcriptional repression can be perpetuated through mitosis, simply because these cytosines are always adjacent to a guanosine residue. CPG island, the phosphate in between probably helps in pronouncing better, otherwise I would say CG. So, CG repeats, normally people call CPG repeats. CPG islands, meaning that in the chromosome here and there you have a lot of repeats of CPG, and that is called CPG island. And these are recognized by a methyltransferase called DNMT3. This does not need either one of the two C's that you see here. See, CG means the opposite strand will be GC, right. So, you have C in both the strands due to this base complementarity. CG means in the opposite, for that G it has to be a C. So, here neither C's are methylated, and such sequences can be recognized by this methyl transferase 3, and that is why it is called de novo methyl transferase. It can start from no prior information for that, and it methylates that. Now, you have a perpetuating methyl transferase that you need to remember, this methyl group is not erased during mitosis, it is going to remain there. Now, after replication, one strand will have the cytosine methylated, the other one will not have, and this one recognizes such methylated cytosines, and they methylate in the opposite strand, the nearest C, and that is how the adjacent G becomes crucial for this. So, now it gets methylated. So, now both strands are methylated. Now, you undergo DNA replication, then one strand will be methylated, another one will not be, and this DNMT1 will do the methylation of that. So, this is how this repressed state can be maintained during cell divisions. So, early on in the embryonic development, at some point you inactivated by methylation, let us say the transcription factor cascade and chromosome modifying, they ended up methylating the DNA. Now, all the cells descending from that original cell, they will all maintain that active or inactive state. Okay. So, that is how this works, and so this has a lot of significant consequences for many situations, particularly here if we look at this dosage compensation. Okay. So, what is dosage compensation? So, like for example, in mammals like human, if you take, females have 2 X chromosomes, males have only 1 X chromosome, while the Y chromosome does not have many really vital genes, but the X chromosome has a lot of important genes. Okay. So, now will the females have one extra load, like will the protein produced from them be double that of males, and will that not cause a problem in terms of the phenotype, right. So, that has to be taken care. So, that is usually taken care by one of 3 different mechanisms. Like for example, if you take C. elegans, both the X chromosomes get reduced by half, and therefore you have the final quantity is like 1 compared to the male that will have only 1 X chromosome. In Drosophila, what happens is the male single X chromosome is doubled up. Okay, its chromatin is modified such that the nucleus, it's truly euchromatin, and the output is more efficient, and therefore it is doubled up. And in human we do the opposite. One of the 2 X chromosomes in the female is totally converted into heterochromatin and repressed. Okay. And that is what you are seeing in this cell. It is a human derived cell where this arrow points to a large black region, and that is the condensed inactive X chromosome. And this is from a person with 3 X chromosomes, and therefore you see 2 black things. These are called bar bodies. Okay. So, that is how the inactivation works. Okay, what is the big deal for me now? The big deal is if you look at the B and C, and if you look at B, what you have is it is a very early embryo. In this you have the reporter LAGZ fused to the promoter on paternal X chromosome. Okay. So, LAGZ will be expressed if the paternal X chromosome is active. Okay. Otherwise, there will be no LAGZ, and therefore this blue color will not happen. So, the pink cells are simply where the paternal X chromosome is not expressing, it is not working. So, this is very early, you see most of the cells having this color. So, this is the inner cell mass from which the entire embryo develops. But when you go to the later stage, if you look at it here, in these cells you do not see the laxate expression. So it turns out that in mouse the trophoblast cells preferentially inactivate the X chromosome of paternal origin, but in other region you can clearly see both kinds mixed. Okay. And there are three important points to remember about this inactivation. One, this starts early in the embryo, okay, meaning in early stage in one cell if it is inactive and then you have an entire tissue or a part of a tissue derived from it, all of that will not have that gene expression. Okay, but if it is paternal, paternal expression will not be there. If it is maternal, if maternal is inactivated, then that will not be there. And second, it is random in which cell which X chromosome, which meaning I am referring to maternal or paternal, is going to be inactivated is random, and once done it is irreversible, it remains in all the descendants of that lineage, and due to that you can have patches of variations, okay, in the somatic body, and that is often very readily visible in organisms where you have skin color that is having patches. Okay, you can see this in calico cats, they have that issue. We saw that in the somatic cloning experiment, and that is how those variations develop. Okay, so these 3 points you need to remember. It happens very early and it can happen random, and once happens it is irreversible. All the descendants will have that inactivation. So if this is the case, then if I have a gene on X chromosome and that is very vital, and if that gets inactivated in my father's genome, let us say, that means having one wild type copy from my mother is not going to be enough. Okay, for certain genes I need my mother's copy for sure, mother's allele is required, and similarly for some genes I for sure need the father's copy. So this is where you will find that the when you are drawing Punnett square, right, it does not matter where the allele comes from, right, maternal or paternal does not matter. But there are situations where, like this X chromosome dosage compensation, where that matters. So we will see that in the next set of slides. Okay. Whatever methylation pattern that exists, they get all of them get erased during gametogenesis. Then new methylations happen. This does not happen to all genes, by the way. There are certain genes that are methylated, and those genes are going to be methylated in one way if the gametogenesis is happening in a male body, happens in another way if it happens in the female body. For example, certain genes may be methylated during spermatogenesis only, and certain other genes may be methylated only during oogenesis, and usually they are mutually exclusive. Genes that are methylated during oogenesis are not methylated during spermatogenesis, and vice versa. Okay. That is what we call as genome imprinting. So simply, what is genome imprinting? Sex specific methylation, as a consequence sex specific expression. If that particular gene is methylated during spermatogenesis, it is not going to be expressed in the from the paternal allele in the offspring. If it is methylated during oogenesis, that particular gene is not going to be expressed from the maternal allele. Okay, so you have two alleles, assume both are wild type, it does not matter which one is inactive. As long as one is definitely going to be inactive, then dosage compensation will not come into picture, right, because one copy only you are going to have. So that is how this works. And as a result you will have to have both alleles. Okay. Like for example, you take gene A that is inactivated during oogenesis. Now even if you have a wild type allele, it is not going to be useful, so you have to have the paternal allele, because only paternal is not methylated, and as a result it is going to be expressed from there, and the same logic holds in the opposite direction too. So the germline uses very different set of combination of the existing molecular biology rules in taking care of its genome. Okay, you can't readily mess with its genome, and the way it protects has its own idiosyncrasies. Okay, and one of them is this, erasing all the methylation and the newly methylate, and the new methylation is going to be based on whether you are a female or male. Okay, so you could have got that particular base pair sequence could have come from your mother. Now if you are male, you will have a male specific methylation added to that gene which originally came from a maternal like a female source, and vice versa. So the methylation is sex specific, sex of the individual in whom the gamete is forming. So now as a result, for such genes, imprinted genes, imprinted meaning sex specific methylation it is subject to, for them you need to have both the alleles, because one allele is going to be inactivated and the other is going to be the only one available. So if that is the case, if you have a mutation in such genes, then you will have sex specific phenotypic outcome. So that is what we are going to see next. So this is a cartoon, so the next will be an actual disease. So here if you look at it, this IGF2, this is not transcribed. So here is a situation where there is no methylation but a factor binds there. Okay, so this binds to unmethylated DNA. Remember in the previous case, like several slides ago, we saw a situation where the activator binds to unmethylated DNA and the activator does not bind to methylated. So here what we are finding is methylation inactivates. So this IGF2 will not be produced when this is not methylated. Okay, that is the case in the maternal genome here. This is methylated in the paternal chromosome. Like what it means is this locus gets methylated during spermatogenesis, not during oogenesis. Now you have an insulator protein binding. So these are the proteins that bind to those silencers, and that binds and insulates this coding sequence from the effect of this enhancer. So this just happens to share the same enhancer, so we need not worry here. So this is produced but not this, this restricts that. But in the sperm derived chromosome, you have a methyl group there, as a result this insulator does not bind and this enhancer activity impacts transcription from IGF2 and that gets expressed. So you need to have both copies. If you had this mutated, okay, like the father carried the mutant version, then you will have, due to mutation you will not have IGF2 protein, and your maternal, although has a wild type copy, it is not expressed. So due to that you are going to be deficient of this protein, and that is it hypothetical? No, it does have real life consequences. So you take these two syndromes, Angelman syndrome and Prader-Willi syndrome. Okay, these are two different phenotypes, coming from mental retardation and seizures and so on. Both have defects. One is the Angelman syndrome is more severe than the Prader-Willi, but double is lethal. So if you look at the first one in this Punnett square, you have the chromosome 15. This is coming, let us say, from a wild type sperm and a wild type egg. You are totally wild type. Now let us say you have a particular region in 15 is deleted in the male. So now you have a wild type copy of that chromosome. Chromosome 15 wild type allele is coming from the egg, but that is not helpful because the required genes are actually inactivated due to maternal specific in imprinting, and when you do not have that from the sperm, then you get this particular disease. Okay. It is because of those particular genes that normally gets expressed from the paternal copy. Now if you look at the opposite, where you have wild type chromosome coming from the male but a deletion coming from the female, a different set of genes are affected because the methylation is sex specific here, and now the corresponding alleles in the male are inactivated, and you need it from the maternal copy, and that is not available due to the mutation, and due to the difference in the genes affected, you get Angelman syndrome. Okay. So this locus is shown in the next cartoon. So this is a complex locus where you have several genes there. The grey indicates inactivation, except here this is a typo, okay, or in the drawing whatever error they had in the book. So this is active in male. So you have, due to methylation, this particular one activates all these genes from the maternal chromosome, but not from the paternal, from the paternal, but in the maternal, due to this methylation, here you have a block of that, and due to this these genes are not expressed. So one set of genes, for example in this particular case, this one is expressed only from the maternal copy, while all these blue ones are expressed from the paternal, and when you need both the gene products, then you need to have both the alleles, okay, maternal paternal both alleles, and this is why you get those diseases. So this is what is genome imprinting, which is a consequence of sex specific methylation. The key you need to remember is existing methylation marks are erased during gametogenesis. Okay. So right now in your cells you will have imprinting, okay, the paternal allele will know I am paternal allele because of a paternal specific methylation. Maternal allele will know I am from maternal because of maternal specific methylation, or the lack of in both cases, and when both are there it is fine. Now in your germ cells, when they are going to enter into gametogenesis, these marks are erased, okay, reset button hit and no methylation. Now if it is spermatogenesis, you are going to methylate a certain loci, and or if it is oogenesis, then you are going to imprint again certain other loci. These are mutually exclusive. So I am stopping here. If you have questions, feel free.